see a lot of marine debris on monk seals and coral reefs, and I just wanted to bring up that we're also seeing marine debris on dolphins and whales out there. This is a rough-toothed dolphin off of Kauai that was actually, I think, playing with this piece of netting. But the fact that it was playing with it indicates that whales and dolphins often like to explore things that they find in their environment, and sometimes the outcome of that exploration is not particularly good. This is a Blainville's beaked whale from here off Kona, back from 2021, wrapped in this cargo net. And in this particular case, back up, see if it comes up better. In this particular case, the cargo net's wrapped around the rostrum, and this animal, I am positive, did not survive. Last month, off of Lanai, we encountered a false killer whale entangled in marine debris, and in this case, it was a loop of floating polypropylene line. False killer whales are often hunting mahi-mahi associated with marine debris, and I think that's probably how this particular individual must have got entangled in it. And you know, this animal hasn't been seen since. Apologies for the graphic nature of the photos. It hasn't been seen since. The next one is even a little bit more graphic, although we're hoping that it'll survive. Now, the reason why I bring this up is because there's at least 3 different organizations here on this island that are doing marine debris removal. Keep Puako Beautiful since, I believe, 1996 has been doing beach cleanups. Hawaii Wildlife Fund has been doing beach cleanups since 2003 on island. And the Ocean Defenders Alliance has been doing cleanups on this island since, I believe, 2017. When we're out in the water, if we see anything that we think is an entanglement risk to a whale or a dolphin or a seal, we're picking it up and bringing it back with us. I know a lot of people who work on the water here do the same. A lot of people are involved in these organizations or volunteer with these organizations, but I just want to give a shout out to all of them because marine debris entanglement is something that I think is actually increasing. National Marine Fisheries Service Pacific Island Fisheries Science Center provided data to me on spinner dolphin entanglements in marine debris, and there's at least 15 cases in the last 10 years with spinner dolphins entangled in debris that had a high likelihood of leading to mortality of those animals. So, anyways, that's one of 2 public service announcements. The other one's going to be at the end of the presentation. So now getting a little bit more to the science and the subject of my presentation here today, and I've given a number of talks on island over the years and Hawaii over the years, and this is my first ever talk on spinner dolphins in Hawaii. I gave a similar talk a few months ago in Oregon, but this project is something that we just really got underway earlier this year. It's a collaborative effort with Randy Wells and Aaron Barleycorn from the Sarasota Dolphin Research Program in Florida. And Colin Cornforth is a major player who's here, and a few other people that work with us. I want to acknowledge that a lot of people have participated in the field work, particularly off Oahu, where most of the field work I'm going to talk about has taken place. We've got a lot of follow-up photos on the tag dolphins from Wildside Specialty Tours, Dolphin Excursions, and Kinwood. Danny Barrios produced the tag animations, which I'm going to show you. Michaela Cravifield assisted with the tag data. And the spinner dolphin tagging work was primarily funded by Dolphin Quest. They gave us a grant to purchase the tags and supported some of our field work off of Oahu as well. Now, going way back in time, spinner dolphins were actually tagged here off Kona in 1971 by Ken Norris. And back then they were hoop netting dolphins, bringing them on board, attaching a fairly large tracking package to the dorsal fin. I'll have a photo later that shows a very similar package of what was being used back in 1971. They caught the spinner dolphin up here, not that far from where we are, and tracked it just by hand from a boat with a VHF radio antenna, and this shows the movements of that animal over a relatively short period of time. At the end of that decade, Randy Wells was involved with Ken Norris and in the Worsigs on 3 more cases where they were capturing spinner dolphins and radio tagging them. and it shows the movements. One of them they tagged over 6 different days, the other just over a couple days. And that was really the sum total of what was known from spinner dolphin movements based on tag data until very recently. Now, I started working in Maui in 1999, and when I came, one of the things that we were doing is putting suction cup tags on a couple different species of whales and dolphins, including pantropic spotted dolphin in this photo, and we tried putting them on a spinner dolphin several times, and they did that spinning thing, and the tags came off. So we basically concluded that for suction cup tagging spinner dolphins was not really a feasible
not a good idea.
approach. Now, some work by Kim Andrews, a PhD student at the University of Hawaii at the time, and I see at least one person in the audience who's a co-author on this paper, using biopsy samples and genetics, basically documented that there's a lot of population structure of spinner dolphins here in Hawaii. And what that means is that the spinner dolphins off Niihau are genetically differentiated from the spinner dolphins off Oahu and the spinner dolphins off Hawaii Island. And in 2011, the National Marine Fisheries Service basically used this genetic information to recognize these 3 different stocks around the main Hawaiian Islands and two separate stocks around the northwestern Hawaiian Islands. There was a report from 2011 by a large group of people, and I'm just seeing if Susan is also a co-author on this one, that used the photo identification data that was available through 2010 to look at movements of spinner dolphins between and around islands. And it has a large group of co-authors because they were trying to compile everyone who had any photo identification data of spinner dolphins at the time. And that study basically found that spinner dolphins generally stayed off the island where they were photographed, but there were some inter-island movements. So there's one individual that moved from Niihau to Oahu, 2 from Oahu to Hawaii Island, and 1 from Lanai to Hawaii Island. So some movements occur, but the movement rates among islands are slow enough that it results in the populations developing these genetic differences among the islands. I will note a paper that was published a couple of years ago from Waianae that only 30% of the spinner dolphins in the Waianae population actually have distinctive markings on the dorsal fin, and that's a real impediment to being able to do those types of photo ID studies to look at movements, because you're at best only going to capture the movements of 30% of the population. And by comparison, in some of the other species we work with, bottlenose dolphins, false killer whales, pygmy killer whales, it's closer to 70 or 75% of the individuals that have distinctive markings. So there's been a lot of research on spinner dolphins in Hawaii, and this is just a sample of studies that are all in one way or another management related. A lot of it's focused on the interactions between spinner dolphins and humans, of course, with the swim with interactions that have occurred both from people from shore and people on boats, and the effects that may or may not have had on spinner dolphin populations. There's some evidence that the population of this island has declined.
And Lars is on it too.
The work, particularly by Julian Tyne, showed that dolphins off of Kona were subject to or exposed to human activities 82% of the daytime within 100 meters. So it tells you the time they concluded that spinner dolphins in Kona were probably the most exposed population of whale or dolphin in the world to humans. It's very little time alone. And these types of management issues have generated a lot of interest in estimating abundance. And the abundance estimation work is complicated by the fact that you have to make some assumptions of where the spinner dolphins are actually spending their time. Or if you're going to survey it off Waianae, or if you're going to survey off Kona for a week or 2 weeks, are the dolphins that you're seeing in that week going to be there the whole time, or are they moving around the islands rapidly? And depending on the answer to that question, it really influences the types of analytical approaches that are taken to do the abundance estimation. So, and I'll just, sorry, I got ahead of myself. we were involved in a study just after a no swim with rule and a no approach rule went into effect here off this island. And even with the rulemaking that went into effect a few years ago, there is still a fair amount of swimming or interacting with spinner dolphins, although it's I think largely now by shore-based launches and shore-based swimmers rather than boat-based interactions. So I'm going to step back a little bit and talk a little bit about tags and tags for different species, whales and dolphins. The image up here, that's from, let's see, 1982. That's the type of tag that was being put on spinner dolphins when they were being captured in the 70s. A fairly large tag that takes up a large portion of the dorsal fin. Now, our collaborators from Sarasota Dolphin Research Program, Randy Wells and Aaron Barleycorn, are both the co-authors on this paper, which was published in 2013, and it talks about development of a new type of tag for use on smaller species. And it's called a single pin fin mount tag. I have one of these tags here if you want to take a look at it later. And this version down here is more or less the same as the version of the tag that we're using today. For many, many years, these tags were only deployed by capturing the animals and supporting them out of the water and then basically attaching them. The tags are attached with a pin that to dissimilar metals, corrodes, and will fall off, or some of them will migrate out the trailing edge of the dorsal fin. Now, Randy Wells and Michael Moore from Woodsville Oceanographic Institution really wanted to figure out a method to deploy these tags on free-swimming dolphins rather than ones that we had to capture. And so we actually worked with Randy and Michael back in 2018 on the very first beta testing of this tool. It's called a tag attachment device, or a TADpole, and I'm going to show you some videos of its use later, but the basic components are these two guides here that basically come around the trailing edge of a dorsal fin that's bow riding, the dolphin's dorsal fin that's bow riding. This is a little pneumatic projector, and it basically has a trigger right in here, and if the trailing edge of the dorsal fin touches that trigger, then in 20 milliseconds it'll insert a pin through and retract and leave the tag attached to the fin. Wow. When we tried it out here in 2018, it turns out, well, the system then would take 40 milliseconds. Turns out dolphins can move faster than that. So the dorsal fin would touch the trailing edge of the dorsal fin and the dolphin would get out before that trigger. So they went back to the drawing board and came up with new electronics to try to reduce that time interval. And I've got one of the pins that is illustrated there that you can take a look at later as well. So they had used this system in the Gulf of Mexico on Atlantic spotted dolphins last year and early this year, and had success. They were able to get 8 tags out on Atlantic spotted dolphins. So with funding from Dolphin Quest, we got Randy and Aaron to come out, and we all met on Oahu to test out this system there. Now, spinner dolphins, this tag system could be used in a number of different species, any species of small dolphin that bow rides effectively. But spinner dolphins, we chose to work with them because of the high management interest, the efforts to estimate abundance, and these assumptions that I mentioned that were sort of untested as to how much time are they spending off Hawaii, for example. Are they there for weeks at a time or days at a time? How quickly are they moving around the islands? We had 9 tags that we were hoping to get out altogether. One of the things that's important for satellite tagging here in Hawaii is that the tags, when they have a saltwater switch, when the animal comes up to the surface, it transmits. And the big question is there a satellite overhead at the time? These are polar orbiting satellites. There were 8 polar orbiting satellites when we started the effort in the beginning, or in the middle of June. By the end of the summer, it was down to 5 of them. NOAA decommissioned 3 of their satellites. But there's a little note at the bottom that we were hoping, and I'll talk a little bit more about it later, that some new nano satellites were going to be functional sometime during the summer. We have 3 different types of tags. A tag that just gives us Argos locations, which basically, if it transmits several times while that satellite is overhead, and it may take about anywhere between a couple minutes and 15 minutes for the satellites to pass over Hawaii, then you can get the locations from Doppler shift. 2 of the tags also had a GPS unit, and it's called fast flow GPS, that basically, in a fraction of a second, it takes a snapshot of where all the GPS satellites are in the sky, and then later on, you can use that to calculate the location much more accurately. That one had the Argos locations 8 hours a day, and the fast flow GPS location 16 hours a day. And then 2 of the tags were what we call splash tags that also record information on diving behavior and give us a lot more behavioral information. So this shows the tool on the end of a long pole, and there's just a GoPro camera mounted on it. This is Makua in Waianae. Lots of spinner dolphins pretty much every day, very shallow water, very interested in bow riding on the vessel, and really just very calm conditions, just ideal situation. So I'll show you another clip that's actually going to show this time a tag deployment in a second here. And that's how fast it happens.
Wow.
So it's very fast. It requires great eye-hand coordination and a certain amount of luck. This tag here is left on the animal. And so this is a photo of one of the tagged animals a couple months afterwards. One of the things we're trying to do is understand the effects of the tags on the animals. Spinner dolphins spin, and that spinning caused an unintended consequence, which is we do get some abrasion on the trailing edge of the dorsal fin behind. I think it's important, though, to keep in mind the types of stressors or impacts from other things in the environment that spinners are dealing with on a day-to-day basis, and this is relevant to their healing capabilities. This is actually a melon-headed whale that has a cookie-cutter shark bite wound right through the dorsal fin, and we see these on spinners. I had a number of spinner dolphin photos. Well, actually, this is a cookie-cutter wound right here on the flank of the animal. I had a number of spinner dolphin photos with fresh cookie-cutter wounds, but they were more graphic than those entangled animal photos I showed earlier. We also see all these nice little remores on spinner dolphins, but it turns out that spinner dolphins and all the other dolphins don't like remores. The reason why spinner dolphins spin is I think, largely because of remores. And, sorry for the graphic image, this is what we call persistent remora damage. So it's a remora that is basically working away at that area just below the dorsal fin. This is a spinner dolphin, over and over and over again, which just shows you why they spend so much time trying to get rid of remoras. If you really want to see something incredibly graphic, Cascadia YouTube page, we have a video of a very large remora on a bottlenose dolphin, and it's basically on that exact same spot. They like that spot, I think, because it's hard for the dolphins to get rid of them there. And you know, the sucker on remora is not a sucker; it's a modified fin that has spines and it digs into the animal. And the video shows the remora actively feeding on the dolphin, digging away and then feeding on the bits of skin that are taken away. So, you know, it shows you that these animals are dealing with these types of transdermal impacts and surviving from them. They do have amazing healing capabilities. Now, spinner dolphins spin, and we were really expecting that maybe we would put 1 or 2 tags on spinner dolphins, and then we would decide that their reaction was too strong because they would just spin and spin and spin. Maybe they would spin until the tags came off. The first one we tagged, it did not spin. It just quickly dove away. The second one did, and of the 9 that we deployed off of Oahu, 5 of the 9, their reaction to tagging would be to spin. 5, 3, 2, and about 15 spins. So it's quite variable. We did tag 1 more spinner dolphin last month off the 9, and it didn't spin. So half and half of them spin and half don't. And I think that the difference between those that do and those that don't could be personality differences, or could have something to do with how much they have been bothered by remorse recently. And if they haven't been bothered by remorse, I'm not sure if that would lead to more spinning or less spinning, but it was quite surprising to us that only half of them spun. Now you would ask what was left on the fin afterwards. These are the photos of the 9 animals that we tagged off of Oahu. And even though some of these dolphins were not what you would call distinctive spinner dolphins, most of them had few, if any, notches, because of the tag placement both vertically on the fin and horizontally on the fin, it was very easy for us to identify all the individuals after tagging. If the dolphin didn't react quickly, then the tag would be, the V part of the tag would be basically flush against the fin. But in the case of that one in the top middle, where the tag is much closer to the trailing edge of the fin, that dolphin was just a really fast dolphin. And between the time when its dorsal fin trailing edge hit the trigger and when the pin inserted, it had already moved a little bit further forward. Now, earlier, one of the slides, I think it said that one of the reasons why this particular type of tag is, a couple reasons why this type of tag is better than many other types of tags. One is the tag can only be deployed on the dorsal fin. It can't be accidentally deployed anywhere else on the bottom because of the design of the trigger mechanism. If a tag is on the dorsal fin, the signals to the satellites are much better than if the tag is at the base of the fin. And the attachment durations from tags on the dorsal fin from that earlier study I mentioned is much, much longer than the limpet tags that we've been using with other species. So you have much less risk to the animal because it's only going to be on the dorsal fin. You have better data coming through Argos and it lasts longer. So we've deployed the limpet tags, which are deployed by an air rifle, on a number of different small odontocetes, bottlenose dolphins, melon-headed whales, pantropical spotted dolphins, pygmy killer whales, and rock-toothed dolphins. And these show the average duration is in this horizontal bar. And over all of these deployments, it's sort of in the range of 12 to 15 days. But our fin mount spinner dolphin tags off of Oahu, the average duration was 42 days. So that part of it was very successful, much longer attachment durations. And in terms of the data that comes through, this shows just a map of each of the 9 individuals we tagged off of Oahu. We tagged them all over about a 7-day period, including 3 tags. The first 3 tags, 1, 2, and 3, were all tagged in the same day off Waianae. The blue dot is showing you where they were tagged. 2 of them were tagged in the same group. Number 2 and 3 were in one group, and 1 was separate, I believe. And what was fascinating very, very quickly from the tag data is to see that the groups that we were tagging were not staying together. They were splitting and joining on a very fast time scale. They were not spending a lot of time off of Waianae, at least some of them were not. And so number 8 down in the middle at the bottom spent a lot of time off Waianae, but the others are spending a lot of time with other parts of the island. And we had 2 that, 2, number 1 and number 7 that went over to Molokai. Now, if you add all those maps together, it basically shows that they're using the entire island. It was quite surprising to me, anyways, that they rest during the day in these areas here and here that are really quite windy most of the time. And people have this idea, including spinner dolphin researchers, that spinner dolphins seek out these protected bays to rest. And that may be the case when there are protected bays to rest in, but that doesn't mean that they're preferentially using parts of the island that have a lot of these protected bays. They're using all sorts of areas. And I had mentioned earlier on when talking about the polar orbiting satellites, and that it went from 8 down to 5 during the study. Well, those other satellites, it's a Kineas nanosatellite constellation that went up. They were actually all up there in July, but we weren't getting any information on what they were receiving until October 1st. So on October 1st, they all of a sudden gave everyone who had satellite tags on anything over the summer access to these data. And this shows the same individual in 2 different weeks in the 2 different data sets. So what I call the legacy Argus satellites in blue on the left, and then the new Kineas satellites on the right. And you can see a couple things. One, there's a lot more locations from that same period. There's just 1 week each. But also, there's a lot fewer locations on land in the new satellites. So they're spending less time on land, or maybe the locations are a little bit more accurate. And some of the locations, so these are unfiltered locations. We go through a process to basically remove locations that are not biologically realistic, like ones on land. But also, in the upper left there, you can see that one location that's way offshore, and that's also not realistic. The same kind of error from Argus satellites that can put a location on land can also put one out in very deep water. And for that same time period, on the right set of maps, that lake location goes away. So, needless to say, we're very happy about this new Kineas nanosatellite constellation, both for the number of locations and the apparent quality of the locations or accuracy, but also the new satellites are overhead 24 hours a day. Before, there were satellites overhead about 14 hours a day, so there'd be these long gaps in where animals were going. The fast lock GPS tags that I mentioned were one way of trying to solve that. But now we've had to reprogram all of our satellite tags to take advantage of the fact that we can potentially get locations 24 hours a day. Now, how many people have seen this graphic? It seems like some people should be putting their hand up that aren't. So this is a graphic that NOAA has produced that basically describes the daily pattern of a spinner dolphin. They spend the day resting in shallow nearshore waters and the night out in deeper water foraging. And I'm going to show you, I think I have 4 different animations of movement data. This one, the yellow is during the day, the black is at night. And they're just some of the examples of what these tagged spinner dolphins did. This one is one of the ones that has the fast look GPS, so we do in theory have data 24 hours a day from this one. You can see it's resting not in these shallow bays during the day really, but further offshore. And I don't know if you guys have spent much time over off that part of the water, but it's not calm there. And then it's moving out and using these habitat features for foraging at night.
That's Cascadia Point, right?
Yes, it is. And that's Makua where it is right now, resting where we tagged a number of them. I should have put music to. But you get the point. So here at night, it's actually very close to shore. Every day appeared to be somewhat different, and every dolphin seemed to be doing something different as well. So there was a tremendous amount of variability. Now, the dive data we were receiving from tags, only 2 of the tags were these splash tags. They were recording any dive below 5 meters depth and 30 seconds in length. And we wanted, again, going back to those surveys, one of the assumptions of aerial surveys is it's basically looking at detection probability. If you fly a plane over, what's the probability of detecting a group of spinner dolphins that are there? And that really depends on how long they dive and how deep they dive. If they dive only a minute, your probability of detecting them is high. If they dive 3 minutes or 5 minutes, the probability of detecting them is low. So that's what we were trying to capture from this. We had 2 different individuals that had these tags, and that graph basically shows the distance between the 2 different individuals in kilometers. And most of the time, they're separated by anywhere between 25 kilometers and 75 kilometers. So the point there is that these dolphins weren't doing the same thing. They were on different sides of the island while we're getting the behavior data, which is great. It's better to get more diversity in what they were doing. And the red and blue bars is when we actually got the dive data. There's often gaps in the dive data, which is why there's white spaces between them. So one tag, we ended up with just over 5 days of dive data over a 13-day period and obtained 1,700 dives. 700 of them at night, and over 1,000 during the day. The other one, we had 9 days of behavior data, 3,500 dives, 1,300 at night, and 2,100 during the day. So I'll show you an example of daytime dives. Now note the depth scale on the left-hand side, just down to 30 meters. So most of these daytime dives, not surprisingly, because most of the time they're in shallow water during the day, were relatively shallow. So between 10 and 25 meters. But the dive durations were longer than we expected. Maximum dive duration in the day of 9 minutes for one and 7 minutes for the other. And so this type of information can then, as I mentioned, feed into understanding the detection probability. Nighttime dives, on the other hand, if you look at the scale over on the left-hand side, now down to 300 meters, were not surprisingly much deeper, and so also much longer on average. The median duration is now 3.6 minutes or 3 minutes compared to 1 to 1.5, although the maximum durations are similar. So they're regularly diving down. I should have asked at the beginning how many people, or how deep do you think spinner dolphins dive? Kind of cool to see what the stats would be. But the overall trends, and this is a 24-hour period in the upper left, and then the entire record in the bottom left. And you can see that during the day, they're primarily up near the surface, and at night they're just diving repeatedly, more or less all night long. And those black bars above are where there's gaps. So it wasn't like it was at the surface there in that one day. It was just there's no data. And that pattern was consistent over the entire record. Shallow daytime dives, deep nighttime dives. These two plots on the right show the average dive depth, again in this solid bar in the middle, and the range in dive depths. Much deeper at night than during the day. At dusk, there are some of these deep dives, and at dawn there's some of these deep dives, and that's basically the animals preparing for or anticipating the deep scattering layer that comes up to the surface at night. And the dive duration is again much longer at night than during the day, and intermediate at dusk. So now a couple more animations. This time it shows 3 different individuals. These are the 3 that were all tagged on the same day, our first field day. And you can see a couple things that I mentioned earlier. One is sometimes they're actually very close to shore in the middle of the night foraging. Sometimes they're fairly far offshore in the middle of the day before they start foraging. Some of that is actually heading out to get started, but other times I think they're just spending time in deeper water during the day that we wouldn't really expect them to be foraging. Here's one that's foraging on the south shore at night, and then still there in the morning. So it's moving out after sunrise, actually, into deeper water. Sometimes they join, sometimes they split. I mentioned that they did start all in largely the same area at the same time.
What is the color mean?
The yellow is during the day and the black is at night.
And what is the link to the bottom?
That is mostly related to the travel speed. So the yellow or black circles are effectively there for a certain period of time. So if the flock gets long, it means it's moving fast, and if it's short, then it means it's moving slowly. And I think that animation may have moved, but it's hard to tell. Here's 2 more, faster animation this time. Again, comparing the day and night for the 2 different individuals, just showing the range of movements, sometimes in deep water during the day, sometimes in shallow water at night. These are the 2 that went over to Molokai. Tag 1, Tag 7, they were tagged 5 days apart, but they joined up briefly while they were on that Molokai run.
Do they sleep?
They sleep primarily during the day. So during the day, they're resting, socializing primarily, although we certainly did see some travel behavior during the day as well. So now I'm actually going to move to a different island. In October last month, we had a project using the same type of tags. We deployed 1 tag on a spinner dolphin off Lanai, And this shows the locations from that tag. This tag is still transmitting earlier today. It was down here off of Kaho'olawe. The main thing I think that's surprising or particularly interesting about this particular deployment is the amount of time it's been spending over Kaho'olawe. For those of you who may be familiar, Kaho'olawe is a closed area. There's no vessel traffic there, which also means there's no researchers going there. And it also shows that, well, I actually got a permit back in 2000 or so, and we surveyed this part of Kaho'olawe and we found spinner dolphins there. But it also shows they're spending time in other areas that you wouldn't generally consider to be relatively calm areas. And as I mentioned, this tag is still transmitting. We also deployed one of these same tags on a bottlenose dolphin off to 90. This tag is still transmitting as well. Bottlenose dolphins, I mentioned for spinner dolphins, there's stock that includes Oahu and Maui Nui together. Bottlenose dolphins are separate stocks that are recognized from genetics and photo identification for Oahu and Maui Nui. This dolphin we actually think is part of the Oahu stock that just spends a lot of its time over in the deeper waters of Maui Nui. And we're still getting data from that one as well. And the reason why I'm here tonight is we just finished up the field project here off Kona. We were primarily looking for false killer whales and beaked whales, and we found false killer whales a couple days ago and beaked whales a few different days. But while we were here, we were also trying to deploy more of these fin mount tags. In this case, this is a pantrop with spotted dolphins. So the first time a pantrop with spotted dolphin has been tagged with this type of tag. This shows the movements of this particular individual. We tagged it just a week ago, and this map is through yesterday morning or so, and it's just moving back and forth up and down the Kona coast. This only shows the Caneas satellite tag data. We are also getting data from the legacy Argos satellites, but it just takes a little bit of work to reconcile those data sets to match them up and filter them. And you can see the antenna of the tag here in the holster. And sometime soon, well, one thing you can notice about this video, I hope no one gets seasick, is that it's not as calm off of this island, despite the big lee, and that was the tag deployment. Now, the dolphin that was tagged actually came in and continued bow riding for another minute and a half. So it dove down quickly, but then came back right up to the boat. So I have one more animation, just because they're so cool. And again, you can see this time it's different colors for day and night for the three different individuals, but it gives you an idea of the coordination of the individuals, as well as the day-night differences in use. Now, these, I mentioned we only got access to the Caneas data October 1st, so we haven't, these are all just sort of preliminary results, graphs and maps to show you what the animals are doing, but it's certainly a very promising technique and is giving us a lot of insights into what spinner dolphins are doing and some of the other species are doing that were just previously unavailable. So now I'm going to go to my second and last public service announcement. I figured I'd pick a subject that is not controversial, and that's cats and toxoplasma. Now, toxoplasma gondii is a protozoan parasite that infects a wide variety of animals, including humans and marine mammals. But cats are the definitive host, which means if there's no cats, there's no toxoplasma. The toxoplasma oocytes, which are more or less the same as eggs, are excreted in the droppings. They wash into streams and waterways. In the ocean, the oocytes can last and persist for a couple of years. And what's important is that the oocytes basically are suspended in water and they're consumed by filter feeders. I only discovered very recently that myctophids, like lanternfish, are filter feeders. I don't really study fish much. And myctophids are one of the main prey sources of things like spinner dolphins spotted dolphins, melanided whales. Toxoplasma can be in an animal and not kill it, so species can test positive for it, but if they have what's called disseminated toxoplasmosis, it's an infection that can kill the animals. Toxoplasma is the most significant disease threat to monk seals here in the islands, with 15 known deaths, and it's the cause of death of 3 different spinner dolphins that is known, which Christy West from the University of Hawaii Health and Stranding Lab has extrapolated out based on the carcass recovery rate to basically reflect more than 60 spinner dolphin deaths from toxoplasma. It's also been known to kill at least 1 bottlenose dolphin, and again, based on the carcass recovery rates, probably is responsible for more than 20 bottlenose dolphin deaths in the last 20 years or so. And a whole bunch of other species have tested positive for it, which include these open ocean species like Fraser's dolphins, other slope-associated species, island-associated species like rough-toothed dolphins, but also humpback whales and rhesus dolphins. And so, if there were no cats in Hawaii, there would be no toxoplasmosis. If there were no feral cats and everyone kept all their pet cats inside and disposed of the waste properly, there would be no, it eventually just died out, because without the cats, toxoplasma can't reproduce. So many of you have probably heard about the Bill 51, which didn't translate well on this. And this is from the American Bird Conservancy in one of their announcements, where they say that Bill 51 is a crucial step towards protecting and restoring native birds, and I added this at the bottom. It's also a really important step for protecting monk seals, spinner dolphins, bottlenose dolphins, endangered false killer whales, humpback whales, and everything else. And the thing about toxoplasma is it's really a dose-response thing. You don't have to get rid of all the feral cats in Hawaii to have an effect, because if you got rid of 10% of them, there'd be 10% less oocytes in the environment, which would mean a 10%, in theory, reduction rate in the infections and deaths of some of these species. So to me, it's one of these issues, for someone who works in a lot of conservation-related science, it's one of these issues that I think is potentially solvable, in part because no one makes money from feral cats. Think about plastics, nets, fisheries bycatch, pollution. All of those things are because of industries where people are making their living from those industries, and so you've got, if you're going to try to solve those types of problems, you've got to work with all of these constituents who are making a living from it, and there's that entrenched interest in being able to continue those activities because it employs people, it's their livelihood. And that's not the case, at least with feral cats. So I think that there's some hope that that particular issue can be not solved, but at least addressed in a way that's going to reduce the risk. At the beginning, my public service announcement was marine debris. I think the efforts of the groups that I mentioned similarly have the potential to reduce the amount of marine debris in the environment, which is going to reduce the threats to all of those species. And I think the same goes with banning feeding the feral cats. So that's my take on that subject. So I think that's it for my slides, and I'd be happy to answer questions.
Yes, so you tagged 9 dolphins, and they did not stay together. But when they were they traveling with other pods?
Yeah, so we actually relocated the dolphins on many subsequent days. So the question was, in case you couldn't hear it, for those who might be watching online, is whether or not the individual dolphins that we tagged were representative of groups of dolphins, and they were. We located, I'm not sure if we located all 9 of them, but we located them. They're always in similar sized groups that they were in prior to tag deployment. And those individuals, many of the individuals have been cited subsequently in groups as well.
The 4 spinner dolphins you mentioned had switched islands. Do you know the sexes on those?
I don't think the sex of the ones that have gone between islands is known. And spinner dolphins are sexually dimorphic. An adult male has a much more erect dorsal fin than an adult female, but I think that report probably doesn't get into that level of detail.
About 20 years ago, there was some work done with a fish finder. Somebody went out with a fish finder at night and reported a cooperative feeding behavior with spinner dolphins. And quite interesting, as you perhaps know. Anything in this work that either supports or refutes that result?
Yeah, that work was by Kelly Baird, who was a PhD student of Whittle Owls at HMD on Oahu, where she, as you said, went out at night with a fish finder and was able to document groups of spinner dolphins foraging together. When we originally decided to tag 9 individuals, my plan was to tag 3 individuals in each of 3 groups so that we could look a little bit at those types of group dynamics. And then what we found out after day 1 is that the groups are splitting and joining so rapidly that it wasn't something that was particularly feasible. We can look at cases when the dolphins were really close together to see if there's any hint of that. Let's say, when dolphins were, say they were in the same group during the day, same resting group, and they go offshore at night, are they staying the same distance apart that they were during the day, or are they twice or 3 times the distance apart? So that's something that we could look at, but we haven't yet.
Thanks.
After applying several of those tags, did you find any effect, any aversive behavior where it got more and more difficult to apply a tag?
Yeah, so again, for anyone online, the question was whether or not after tagging 1 or more individuals in a group, whether other individuals or maybe the same group on subsequent days showed any difference in the bow riding behavior and ease of tagging them. And I would say no. We have no evidence of that. What we found when we tried to tag off of Lanai and when we tried to tag spotted dolphins off this island is that the conditions, the sea conditions, are the biggest determinant of whether or not we can actually get a tag deployed. And so calm conditions are really critical. In rougher sea conditions, the boat makes more noise just moving through the water. And that noise of the boat just hitting against the waves means that even if dolphins are bow riding, they may be bow riding 1.5 feet too far ahead. The animals, we have a pulpit on the boat, the animals more or less have to be straight down. But we didn't see any evidence that the groups were avoiding this afterwards. We had dolphins that we reapproached on subsequent days and had some that bow rode with the tag on in subsequent days. I don't think the rest of the individuals in the dolphin groups really connected that tag and us in terms of their behavior towards us. And back.
May not had enough time for this, but have you noticed an impact on like the omnidirectional sonars or boat traffic on the dolphin behavior?
Yeah, it's a good question. There's been a big increase in omnidirectional sonars in a lot of the larger fishing vessels, sports fishing charter vessels, in particular in Hawaii, in particular off this island, I think. I think that some species of whales and dolphins are very sensitive to those types of sounds and others aren't. We've noticed a decrease in pygmy killer whale sightings off of this island in the last few years, and it's certainly pygmy killer whales are a species that's known to be sensitive to military sonars, for example. I don't know what's causing the decrease in pygmy killer whales here. They're not declining in sighting rates off of Oahu, and I don't know if anyone here knows much about the use of those omnisonars, say off of Oahu compared to Kona. If anyone does, please chime in. But my suspicion is that they're affecting some of these particularly sensitive species.
Is that tagging device attached to the boat or handheld?
So the pole itself is handheld. And we have a scuba tank, so that's what powers it. Well, we have batteries that power it, and a scuba tank that powers the actual pneumatic projector. So someone stands up in the bow. Colin has tagged our 5 of the last 6 animals, and Aaron Barleycorn tagged the first 6 that we tagged off of Oahu, and Mark Moeller has tagged one as well. But they're basically standing at the bow and showing a lot of patience and waiting, turning the camera on and off so we don't get hours and hours of that video footage.
I think it's a lot of physical effort just to hang on to it.
Absolutely. I just drive the boat, I should say.
It's not streamlined, really.
Yeah, well, I should also note that the TADpole itself, these tools, there's only 2 of them in existence. They're individually made by engineers at Woods Hole. When we started the Oahu project, we had 2 functional ones, and partway through the project, one of them broke. So it's not like it's off-the-shelf available technology yet, but it's gone from deployment by one species in the Gulf of Mexico to now 4 different species. There's now 4 different individuals who have used the tool to deploy tags. So it's a dramatic increase. Sarasota Dolphin Research Program just got a grant from Dolphin Quest actually to buy 2 more, or get 2 more of them made. And they are, there's a lot of interest in being able to look at movement patterns of different species of dolphins around the world. And so they're working with a number of different groups. I think their next trip is down to the Galapagos to take the tool down there and try to do some bottlenose dolphin tagging in the Galapagos. And it's not a panacea. We know from experience that there's many circumstances that it won't work. So it has to be cooperative animals in calm seas, and those don't always come together.
How do you know when the tag's fallen off?
It stops transmitting. Well, I should say, I mentioned at the beginning there are 3 different tag types. The location-only Argos, the ones that have the fast talk GPS, and the splash tags. Each one has a different battery life. And so we know of at least 2 cases, although the batteries didn't die until 40 days into the deployment, where the tags remained on after the batteries went dead. But we only know that because people off of line, I got photos of them, they had stopped transmitting. I mean, I checked, here, let me check and see what the tag diamonds are. I mean, I check obsessively on my phone a couple times a day to see. Right now we have the spinner dolphin and the bottlenose dolphin off of Lanai, and the spotted dolphin off this island, but we also have a few other tags out on other species, the limpet tags. And so there's several of us that are checking regularly to see where they are. In the case, and I'll go back to Doug's question earlier about do the animals avoid the boat, we relocated our tagged spotted dolphin out here maybe 3 days after tagging, and it came back in right to the bow at one point. Very bad.
So did you guys learn anything new that you didn't know about movements of what these dolphins are doing?
It was amazing, frankly. The first couple days when we're looking at the movements of the animals, I think it was surprising, not just us, but people who have been studying spinner dolphins for the last 20 years, people in the tour industry in Waianae that watch them every day. I mean, the most striking thing, and keep in mind we haven't actually really analyzed any of the data, we've just visualized it yet. But the most striking thing was the rates at which individuals leave and come back to Waianae. The use of deep water during the day was surprising, I think, to everyone. The use of not calm water during the day for resting was surprising. I think most of us had thought that when they're on the windward side of the island, they go over there when it's calm over there, which isn't very often, but that's not the case. They're spending a lot of time on the windward side of the island. We would like to deploy these tags on spinner dolphins off this island at some point in the future. And, you know, every time we've put one on, we've learned something new. It's on the very steep end of the learning curve at this stage.
Are the tags able to be removed, or do they fall off eventually after the batteries are dead?
Yeah, well, if anyone wants to take a look at this, the tabs of the tag have two different rings. One is Delrin and the other one is metal. The pin that goes through is metal, but it's a dissimilar metal. So what happens is that the dissimilar metals cause corrosion, and eventually the pin will corrode, and the ring on the tab here will corrode, and the tag will fall off. Did that answer the question?
Yeah, I was just wondering about the damage on the dorsal fin, if it continued to be attached, that it would be detrimental to the animal.
We've had two of them that have torn through the trailing edge or worked their way through the trailing edge. I think that spinner dolphins, that's going to happen more than bottlenose dolphins or spotted dolphins, just because of the spinning. And you know, we are trying to constantly get follow-up photos from people who are on the water in order to be able to document those types of things. Randy Wells, in their work where they deployed many of these tags on bottlenose dolphins in Florida, you can get situations where the fin falls out and you'll end up with a piercing, a hole through the fin. In some cases, that'll completely heal over, best case scenario, and in some cases, it'll migrate out through the back of the fin.
Over the years, we've heard all kinds of answers or suggestions as to why spinners spin. So are you saying now that the remoras that's the definitive answer?
That's my definitive answer. It's amazing with the availability of cameras that have motor drives that take 40 frames per second. I would be willing to bet that if you had a very good sequence of photos of a spinning spinner dolphin, it's going to have a remora on it.
Yeah.
They actively, you can watch them and they actively will spin in a certain direction, or they'll land in a certain way where they're trying to either shake the remora off, or they're trying to slap the remora off. And I should say, I think this applies to adult spinner dolphins. Juveniles may be doing other things. You know, juveniles of any species are doing these behaviors, not necessarily for some functional reason.
They're just learning how to spin.
Yes, in fact.
So does that mean that the remora generally feed on the spinners? And if not, or if they're feeding on the other type, how are they getting them off?
Well, it's really worthwhile looking at this underwater video we have of this bottlenose dolphin with remora off of Lanai from a few years ago. It's on our YouTube page. That remora is actively feeding on the spinner dolphin. It's moving back and forth and generating clouds of tissue that are coming off the animal, and it's feeding on that. I think that remoras probably also feed on scraps that are left behind from whatever their host. I think some species, not spinner dolphins, but false killer whales, I think they actively remove remoras from their buddies. I think pygmy killer whales might do the same as well. But then there are other species like bottlenose dolphins and spinner dolphins and spotted dolphins that I don't think there's any evidence of that, and they just spend a lot of time trying to get rid of them themselves.
Hey, Robin, I understand the value of the group dynamics and understanding that and where they go and that sort of thing, and looking for habitats. What do you see as sort of the ultimate goal of the program that you're pursuing? Well,
most of the work we're doing is I mean, there's a lot of interesting behavioral questions that can be answered with it, but most of it's driven by management-related questions. And I've reviewed many of the publications on spinner dolphin abundance estimation, and I see the information that they need in order to come up with more definitive answers. So the abundance estimation work, the whole purpose of the abundance estimation is not only know how many animals there are now, but how that population is changing over time. Is it increasing or decreasing or remaining stable? And you know, I mentioned some of the other threats that these animals are facing, whether it's marine debris or toxoplasmosis or harassment, fisheries interactions. All of those things are occurring. The question is, are they causing populations to decline? I should say we published a paper earlier this year on false killer whale abundance estimation and trends, and the paper used an approach that took into account the effort of the researchers versus where the animals spend their time. And this kind of data set of where the animals spend their time fits extremely well into that framework of being able to estimate trends in the population. So in the short term, the purpose of this is to provide input to the researchers that are trying to estimate abundance. But we're going to get a lot of other information as well. I mentioned that there's two ways that people are trying to estimate abundance of spinner dolphins. Three ways that people are trying to estimate abundance of spinner dolphins. One is mark recapture estimation from photo identification. One is line transect aerial surveys, and the other is line transect vessel surveys. Those line transect estimates really rely on this estimation of the proportion of animals, their probability of detecting a group of animals, and the dive behavior data is relevant to that. So the reason why we're doing this and focusing on spinners is because of the inputs that these data sets can provide to those other types of studies.
Thanks.
Going back to the remoras again, do you happen to have any theory for why South Pacific humpback whales in Tonga are frequently just covered with remoras, and North Pacific humpback whales in Hawaii very rarely have a remora or two on them?
I do not have any possible explanation for that. Do you have a theory why? Yeah, it is very interesting. You know, here in Hawaii, some species of donosids have a high incidence of remoras and others have virtually none. I mentioned false killer whales. The only remoras we've ever documented on false killer whales are these little tiny ones. And I think it's because they're too small for another false killer whale to get its teeth around. You know, the big ones they can remove pretty easily, and one of their buddies could easily come over and grab that remora and take it off, and I think that's what happens. But you know, I'm in touch with colleagues in the Azores or the Canary Islands where they see far fewer remoras, and I showed that one graphic image of the remora, persistent remora damage on the spinner dolphin, and they had something like that on an animal in the Azores, and they were wondering what was causing that particular type of lesion on the body. And we, with our photo identification catalogs, like the individual I mentioned that we have the underwater GoPro footage from Lanai, we re-documented that individual a number of times and have looked at the wound healing from it, and you can come up with these very consistent wounds. But again, it's very species specific for even within Hawaii of what species have those persistent remora damage wounds.
Huh.
Remoras being called shark suckers.
Yes.
Is it the same species or are they separate?
Yeah, no, these are probably all shark suckers. Yeah. It's very difficult to identify remoras to species from photographs. My wife has studied remoras a bit, and I show her all the photos we have of remoras on dolphins, and that's the case. I keep hoping that someone is going to collect a remora, and if so, you know, if they found a dead remora somewhere, I'd love to get it. I'd love to get it.
Good luck.
Just put it in the freezer. Amy, it won't go in your freezer. Call me.
Say we can work off this island. This is our local community right here. What do you hope we would accomplish if we could get some of the groups tagged off of this island in the near future?
Yeah, well, for spinner dolphins, one thing I didn't mention, but you may, for people who've been around for a while, remember that Macaco Bay used to be a great place for rescuing spinner dolphins going way back in time. And spinner dolphins have more or less abandoned Macaco Bay. I think that there's a couple possible reasons for why they've abandoned Macaco Bay, and one of which is the fish farm right off and the bottlenose dolphins that associate with fish farm, or the sharks that associated with fish farm. So in the last 10 years, the spatial use of spinners has changed off of this island. Whether or not the Macaco Bay area, either because of the sharks or the bottlenose dolphins, is excluding spinners. What are they doing as they're going by? I think that has implications for at-sea fish farm development elsewhere in Hawaii. Off of Oahu, there's been a couple published papers recently on spinner dolphin abundance estimation. There's one in the works right now for Hawaii Island from Claire Lacey at the University of Hawaii. And I haven't seen those results yet, but I think that the same types of information on spatial use movements around the island would be of value. You may or may not have seen it. I don't know if I pointed it out in one of the animations, and this more gets to a behavioral question rather than a management question. But it's very interesting to me from the behavior of the animals that in some areas around Oahu, that nighttime foraging is very close to shore, and in other areas it's way offshore, and trying to understand the reasons for that, both around Oahu and around other islands, would be of value. I think there's a lot of different questions. Some of them are to better understand the species, and some of them I think have direct management implications as well. And getting back to the abundance estimation very quickly, part of the problem with the abundance estimates is that science changes over time, and people realize that there's better ways to do things. And in the abundance estimates from Julian Tyne's work, compared to the more recent work, compared to the older work, the techniques have improved, the methods have been improved to reduce the biases, but it makes it harder to compare the estimates from one time period to another. And understanding the movements of the animals around the island, I think, would help inform the interpretation of those abundance estimates.
Your comment about foraging, is it known what species these animals primarily prey on?
Spinner dolphins are yes and no. I mean, there's a general idea that they're feeding on prey that are associated with the deep scattering layer as it comes up towards the surface at night. What's known about the diet is really restricted to stomach contents, and there have been a handful of animals that are stranded with stomach contents. Christy West's lab at the University of Hawaii is now starting to, or not now, they've been doing it for a number of years. They've been collecting material from the intestines to do genetic analysis of what the prey would have been. So it's effectively collecting fecal material from the intestines. And I think that's going to be extremely informative to better understand the suite of prey. I'll use this example of melon-headed whales simply because Christie published a paper on melon-headed whale diet from stranded animals. And melon-headed whales and spinner dolphins melon-headed whales are feeding further offshore at night than spinner dolphins, but they're both feeding on the prey associated with the deep scattering layer. And melon-headed whales have a very diverse diet of those mesopelagic squid and fish. And I suspect the same is true for spinners, but the sample sizes of stomach contents is just limited at this stage. I will mention, though, related to the fecal samples, that some of the work that we've done is collecting fecal samples from behind whales for that purpose. I did a lot of work with killer whales in the Pacific Northwest, and one of the studies was following behind fish-eating killer whales, collecting fecal samples, and then they were running the DNA on those. What people would see them feeding on were primarily Chinook salmon, but lo and behold, from the fecal samples, they're also feeding on other things that are smaller and deeper in the water column. So there's a variety of ways of looking at diet. It's stomach contents, direct observations, genetic analysis of fecal samples, and they're all complementary.
So fascinating.
Any more questions?
That is, it's like a bullpen.
Well, I did put some laminated handouts that we have up there, one on bottlenose dolphins, one on killer whales. For anyone who works on the water, the idea is that they're double-sided. It's a way of, the backside, there's information on the biology of the animals, how we're studying them, and on the front side, it's IDs of some of the distinctive individuals, and we made those up for people who dive operators, tour operators, just to be able to have something to show their passengers. So if you work on the water or play on the